ஜாயின் ஆயிட்டாரா இல்ல?
நான் சொல்லிட்டேன் சார், நான் சொல்லிட்டேன்.
அப்படியா?
ஆமா, நான் அவர்கிட்ட சொல்லிட்டேன். வந்துட்டாங்க 2 பேரும் வந்துட்டாங்க.
அப்படியா? ஜாயின் பண்ணி.
ஆமா, டீன் சாரும் வந்துட்டாரு, ஆமா ஹெச்ஓடி சாரும் வந்துட்டாரு.
சார் குட் ஈவினிங் சார்.
Sir, good evening, good sir. Namaste, sir. And
Yes, sir.
Agateshwaran.
Good evening, sir. Shall we start, sir?
Okay.
Okay. Winners don't do different things, but rather they do things differently. With this, we start this final day session. Yes, we are all here together for the final day session of a 3-day international webinar series on unlocking approaches in biological research. Nothing is impossible with our God as long as we have faith on Him, on His mighty power. As we march forward into a new agenda today, with a great hope and aspirants, let us invoke the blessings of God through the prayer song.
The Bible says it's a good thing to give thanks to the Lord. Amen.
Thank you, sir. Welcoming is a part of any academic event which gives proper way of beginning. So I would like to welcome each and every one of you to this final day session of the international webinar. The courage to make tough decision and the compassion to listen to the need of others is an art. This quote is very filling to Dr. S. Agaya Satish, the Dean, School of Biological Sciences. Sir, your kindness, encouragement, guidance, and your positive support prove that you are a great mentor and leader. We are really honored to have you, sir, as a dean for our School of Biological Sciences. With a thankful heart, we welcome you, sir. If your action inspire others to dream more, learn more, become more, you're a leader. At this juncture, I would like to attribute this proverb to our beloved head of the Department of Botany, Dr. S.R. Sendil Kumar. Because of his valuable guidance and the encouragement, we made this international webinar as a successful one. With a great pleasure and a gratitude, I welcome you, sir. Welcome. It's my great privilege to welcome our illustrious alumnus, our today's resource person, Dr. Agatheswaran, Associate Researcher, Division of Molecular Medicine and Gene Therapy, Lund University, Sweden. On behalf of St. Joseph's College Management, Botany and Biotechnology Department behalf, on my own behalf and the participants' behalf, I extend you a very sincere welcome you, Dr. Agatheswaran.
Thank you so much.
Nothing great will be achieved without a great man. A great man with us, our organizer and the head of the Department of Biotechnology, Dr. Edward. I am happy to welcome him on all of them behalf. Welcome, sir.
Thank you, Professor.
With the immense pleasure, I welcome our participants from the depth of my heart. Welcome all of you. Welcome one and all. It's a time to welcome and introduce our today's resource person, Dr. Agatheswaran. So he is a proud alumnus of our department. He did his UG and PG programs in plant biology and plant biotechnology during 2003 to 2008. After his PhD program, he did his doctoral degree Utkal University, Bhubaneswar, Odisha. In his doctoral research, he identified MIR 223 is a down-regulated during CML progression and target transcription factors MEF2C and RNA binding protein. So he has received various awards during his doctoral research and post-doctoral research. Now he is a associate researchers in the Department of Molecular Medicine and Gene Therapy, Lund University, Sweden. So his biodata is a very lengthy one, and I welcome our resource person, Dr. Agatheswaran, on behalf of the St. Joseph's College Management and my own behalf. Welcome, Dr. Agatheswaran.
Thank you so much, sir.
So it's a time for the resource person. So Dr. Agatheswaran, he will share his research expertise with the participants. Welcome, Dr. Agatheswaran, over to resource person. Please, sir.
Thank you so much for the nice introduction. I'll directly get started with the presentation. Hope all of you can see my desktop now.
Yeah, we are seeing.
Perfect. So I can get rid of this now. Yes. So thank you so much for the introduction. and I would like to get started with the presentation. So the title of my topic is Blood Forming Cells, Regeneration for Life and the Evil Twins. Yes, Dr. Francis introduced. I was an alumnus of St. Joseph's College, Plant Biotechnology Department, and I did my master's in Plant Biotechnology. Then I decided to move towards the medical research, because which was my dream back then, even in the school days. So I will go through my journey a bit, then I will get into my research work. So this is the outline of today's talk. I have formulated into 5 major divisions. The first part is I would introduce background about the blood system for those people who are not involved in the blood system. And then I will talk about the disorders of one of the common disorders of the blood system, leukemia, which is commonly also called as blood cancer. Then I would briefly talk about my research interests. Then I will be presenting 2 of my recent studies. The first study is about identifying a novel stem cell marker, and the second study is utilizing novel stem cell expansion approaches for clinical therapy. So this is a brief slide about my CV, which says like I did my master's and then I moved to cancer biology to pursue the medical research. Then I moved to Lund University here in Sweden as a postdoc, and currently I'm working as an associate researcher at Lund University. And the point here is also I wanted to make nothing is too late. So even if you're doing any part of life science, you can shift to any part of life science. It's quite easy, and most of these things are same. So the biology is same, so you can shift at any time you want. So let's talk about blood system. So this is a very basic slide explaining the components of blood system. So majority of the blood contains plasma and cellular components. First, let's talk about the plasma. It contains mostly water and some of the proteins, and of course it has the ions. And the major function of the plasma is taking care of the nutrients delivery to the tissues and cells, and it also collects the wastes and involved in eliminating the wastes. And of course it's also important carrier of many of the chemical messengers like hormones. Then let's talk about the cellular components. It's broadly classified into 3 groups here: red blood cells, which mainly carry oxygen to the tissues. Then we have white blood cells, which are mostly involved in the defense mechanisms, which protects us against the pathogens. Then we have platelets, which are essential for healing the wounds, which is a major function again. But when we look at the lifetime of these cells, it's so less when compared to a human life. Let's see the life of a red blood cell. It's only 115 days. And the white blood cells even less. And for the platelets, which is even less. So an average human lives around 70 to 80 years. How these cells are produced again and replenished? That's the $1 million question back in the 19th century. So the answer comes from bone marrow. In a day, human bone marrow approximately produces 1 trillion new cells, which is quite amazing. And these are continuously produced until a human's life. So it's a lot of work. And previously, theories have been proposed that a common single stem cell present in the apex, and it is responsible for producing all these different lineages of cells. But there was no evidence. It was just existing as a theory until the Second World War. So this was one of the darkest episodes in human history, where the atomic bombing in World War II has killed like 300,000 people. But there was an interesting observation. Majority of this casualties didn't come due to the heat or something. Most of the people died after a week of the bomb explosion. Then people suddenly start researching about the consequences of radiation, which was still a quite new field back then. And what they found was amazing, and it was quite interesting that the radiation released from the nuclear bomb was inducing bone marrow failure, and the people who got the bone marrow failure was living like a week or two before facing their fate. But meanwhile, researchers in 1950s they could introduce the same effect in mice models. When they exposed mice to lethal doses of radiation, they observed the bone marrow was the main casualty, and then they also observed when they transplant cells from similar mouse, and they could rescue the dead. And it was the origin of transplantation, and that's how the field evolved. Then in 1960s, two researchers from Canada, Till and McCullough, they were the ones went on to show that there was a single cell which is responsible for producing all these different lineages throughout the lifetime, and they did this with variety of in vivo repopulation assays. So then the definition for stem cells, especially hematopoietic stem cells here, it came with like this: a cell which can produce blood lifelong, and they can self-renew themselves without getting exhausted while contributing to the differentiating cells. And the last point is, due to this unlimited cell divisions, these cells can accumulate some errors during the DNA replication, which some of which could be deleterious mutations, which could lead to failure of the system, and which is also called leukemia. So how a single cell can produce different lineages of cells, and most of you would have seen this image in any immunology book. This is the product of 50 years of research. People can establish that a hematopoietic stem cell, which is sitting in the top of the hierarchy, can produce a multipotent progenitor, and which mainly goes towards two different arms. The first arm is a lymphoid lineage, which goes on to produce B and T cells. These are the seals of adaptive immunity. Then it also goes on to produce NK cells, dendritic cells, and monocytes. And there is another arm, which is the common myeloid progenitor, which goes on to produce two groups of cells, monocytes and granulocytes, which are responsible for the innate defense in our body. Then this also produces erythrocytes and megakaryocytes, which are responsible for oxygen transport and blood clotting in case of an injury. But the question is how a single cell can stay here and produce these cells continuously. The trick lies on a simple logic. When a stem cell divides into two daughter cells, one of the cell becomes a stem cell and the other daughter cell becomes a multipotent progenitor, and this multipotent progenitor goes on to produce different lineages of cells, whereas the stem cell again goes to take the spot of the previous stem cell. This is how the stem cell stays forever in the human body. However, as I said, the stem cells has to divide quite a few times because they have to produce blood like every day. And in this process, I just briefly put the cell cycle here, which involves the synthesis phase where the DNA is synthesized. But it looks simple, but synthesizing a new DNA is not that easy job because we have like 3 billion base pairs, and even a single error in that process could be deleterious. So some of this errors can accumulate in the stem cells because there is no way, if the error is not getting repaired, it can persist for lifelong. And some of these errors accumulates in some clones, and later the errored stem cell can accumulate secondary mutations, which can develop into full-blown leukemia. Then with chemotherapy and other therapeutic targets, we can still eliminate some of them, but eventually it's very difficult to eliminate them completely because by then they accumulate quite a few mutations, which is very difficult to respond to a single drug. And that's why these blood cancers are so lethal. And if we look at the survival rate of some of the blood cancers like acute myeloid leukemia, the 5-year survival is only like 20-25%. So there is a serious discovery for new treatment methods to treat such disorders. As I said, these deleterious mutations doesn't accumulate in a single site or in a single gene. So I put the central dogma here just to represent how the basic protein synthesis is regulated. And most of these mutations which we observe in any cancers, they are always associated with essential cellular responses like the pathways associated with proliferation or the pathways associated with DNA repair. These are the ones either enhanced or compromised. And some of these oncogenes can also modulate the expression of microRNAs. They can also in turn alter the protein expression, and which could also invent new cellular responses. The point here is, again, I wanted to make what kind of mutations commonly exist in blood cancer. If you see that, it can be mostly, some of them are even quite deleterious because these are translocations between two chromosomes. So a part of the chromosome moves from one region to another chromosome. So this is a quite serious mutation. And there are incidents where inversion of the chromosomes can happen, and there is also quite a lot of incidents where point mutations tend to occur. But as I said before, majority of these mutations occur in chromatin factors and like pathways associated with cell cycle arrest like P53. Hence, the cells can gain proliferative advantage and they expand with unlimited proliferative stimulus. During my PhD work, I was studying a part of leukemia called chronic myeloid leukemia, which mostly happens in the adults. So it's like any other model I have shown before. In chronic myeloid leukemia, a normal stem cell gets a hit where a translocation occurs between chromosome 9 to 21, and as a result, two of the genes, unrelated genes, getting fused, and they form a fusion protein called BCR-ABL. As a result, this single translocated protein can dictate the stem cells to produce only unlimited myeloid cells instead of lymphoid cells. So as a result, the hematopoiesis is severely impaired. But over the past 3 years, there are additional mutations. They tend to occur in these cells. As a result, the disease progresses from a more of a chronic phase to a advanced blastocytosis. As a result, the patient, it's very difficult to treat at this stage, and it's quite hard to save the patient after this. So I was mostly interested in identifying novel molecular mechanisms which are contributing to the survival of these cells. And some of the papers I have presented during my PhD are represented here, but I'm not going to talk about this work today. Today I would be talking about my quite recent research work. So the point I wanted to make here is during my PhD. So I got to understand the molecular mechanisms of leukemia and how serious they are and what kind of novel therapeutic strategies has to be developed. This was my basis of the education. So let's talk a bit about leukemia therapy. Then I will move into my postdoctoral work, which is associated with one of the leukemia therapy. So in this image, I just took it as a common cancer treatment options. So it's not only limited to leukemia, but I can draw some of the inspiration for the leukemia treatment from here. As you can see, chemotherapy, which is commonly used clinically for treating leukemia, but chemotherapy as such is not a targeted therapy. The success of chemotherapy relies mainly on sensitizing the cells which are having higher proliferative advantage than the cells having a lower proliferative advantage. So that's how chemotherapy was successful in eliminating leukemic cells, although it was not exclusively specific to leukemic cells. But then people also started developing more targeted therapies based on the molecular mechanism of the cancer. But as I said before, cancer is, at least blood cancer, is caused by mutations in more than 500 genes, which means developing a targeted therapy for a single molecular pathway could only cure a small group of patients, since all of this look as a leukemia phenotypically, molecularly, they are quite different. So targeted therapies has to be developed for each of these mutations separately. That's one of the caveats. Many of the leukemia subtypes are not having targeted therapies yet. And also another interesting thing is immunotherapy approaches using human immune system against the leukemic cells, which is also in the clinical trials, and their success has to be seen yet. And also there is one long-term cure for leukemia currently available, which is bone marrow transplantation. In this case, the whole leukemic cells and the normal cells are eliminated from the patient's bone marrow, and new stem cells are introduced into the patient's bone marrow from the donors. As a result, the new stem cells from the donor establishes a new blood system. Thereby, the patient can have a long-term disease-free survival. So the remaining talk will be associated with bone marrow transplantation approaches and what we are doing here to improve the bone marrow transplantation. Although, as I said, bone marrow transplantation technique, as I said, it's quite easy to say it's a curative therapy, but there are a lot of roadblocks here. What are they? So this is the outline of transplantation. In this, a suitable donor is identified based on the HLA-MH, which is a human leukocyte antigen, because not two human beings are same. Maybe two twins are same genetically, but no two human beings are same genetically if they are not twins. So you have to identify people that at least have 50% of match with the human leukocyte antigen to become a successful donor. Then the patient's leukemic cells and the immune system, everything is destroyed basically with a chemotherapy regimen. It's called conditioning. Then these cells are introduced into the patient. As a result, the patient can make a new blood system now from the stem cells donated from a donor. And as you see that the engraftment is quite quick and the immune recovery, other things are quite quick. But there is also a problem here called GVHD, which is called graft versus host disease. What is this? So the stem cells derived from this patient, they develop a full immune system, but that immune system recognizes this person as a foreign object, just like because due to still some of the existing HLA mismatch. So that is one of the major caveat in this setup. So the more similar donor, more better outcomes. And what are the sources of hematopoietic stem cells? As I said, the donor could be an adult, it could be a relative or non-relative, but should have quite decent overlap with the HLA expression, HLA sequence. Then there is also recently identified source of hematopoietic stem cells called umbilical cord blood, which is isolated from the umbilical cord of the newborn. Generally, when the child is delivered, the umbilical cord, which is connecting the child with the mother, also carries quite some amount of blood. But researchers have identified that this blood is a rich source of hematopoietic stem cells, and these hematopoietic stem cells are also able to reconstitute if transplanted. Since umbilical cord blood is a medical waste, it's not used for any purpose. It's a quite easy resource than isolating hematopoietic stem cells from a donor. Also, here I have listed the advantages of different sources like bone marrow and cord blood, and also disadvantages. As you can see, in bone marrow, it's more invasive HSC collection because typically the samples are collected from the bone marrow, which is quite painful, and it's quite painful to take bone marrow biopsies and collect samples. But then there was a new method developed where they have identified ways to mobilize the stem cells out of bone marrow. Then the cells are collected from the peripheral blood, but these sources is a non-invasive method, but it's still associated with higher graft versus host disorder. Then comes cord blood. Cord blood is non-invasive. For some reason, cord blood transplants produce lower graft versus host disease, which is not clear why it does. So hence, if you see this HLA matching, probably with the adult donor, it has to be strictly matched. But with cord blood, it can be less strictly matched, which means finding a cord blood donor is quite easier compared to finding an adult donor. However, the stem cell dose are determined based on the body weight. So usually the stem cells obtained from cord blood, which is usually sufficient to reconstitute a child. Let's say in medical terms, pediatric patients can be transplanted with hematopoietic stem cells from cord blood, but due to lower cell number, an adult actually needs quite a lot of cell dose, which makes it harder for using for the adult patients. But however, there is a way if we find to increase the number of stem cells which we isolate from the cord blood. So during my postdoctoral work, I joined Department of Molecular Medicine and Gene Therapy. They work extensively on human hematopoietic stem cells, especially from the cord blood, and to some extent from the bone marrow donors as well. And the group I joined, they were mostly interested in identifying novel mechanisms that can expand stem cells in the plate. As a result, we could expand the cord blood stem cells and transplant to the adult. This is called allogenic transplantation. But optimizing a suitable cell culture condition for the hematopoietic stem cells is also having quite a lot of other medical applications. Like some cases, patients are having point mutations like sickle cell anemia. So their stem cells can be isolated and cultured, and using the gene manipulation techniques, the genetic disorders can be corrected, and the corrected stem cells, gene corrected stem cells, can be expanded, and they can be transplanted back to the same patient. In this case, the patient will not have any graft versus host issue. So this is also one of the major application where people are interested in developing suitable in vitro culture conditions for expanding stem cells. So there are three different fates of a dividing stem cell. First, it can make two daughter stem cells. It can produce one daughter stem cell and one progenitor stem cell. These both are called self-renewal or expansion. However, there is also a worst consequence where a stem cell can produce two sister cells. Both of the cells can become a progenitor. As a result, we could lose the stem cell forever. Initially, when people thought it's quite easy to achieve this, probably with the standard culture conditions, but with the in vitro culture, if we expand them for a week, what usually happens is the worst consequence. So if we culture them for like 14 days, then most of the cells differentiate into progenitors. As a result, it's quite difficult to expand hematopoietic stem cells in vitro. That's the most challenging thing. So hence, for the past 2 to 3 decades, people are interested in developing or identifying novel regulators which can promote expansion of hematopoietic stem cells and preventing differentiation. So I would like to list some of the top findings from this field. They could identify a set of cytokines and growth factors which can support human hematopoietic stem cells in the in vitro cultures. And they also found overexpression of some of the genes, HOXB genes, and inhibiting HDACs, or overexpressing microRNA 125A. They could achieve expansion, but however, the introduction of this transgenes are mediated by virus. So due to the integration of the virus, it could also produce long-term unwanted consequences. So these are not going into the clinical trials. But then there is an alternative approach. There is an alternative approach where they used small molecule compounds like two of the top hits identified were SR1 and UM171. I'm getting echo. So is there a way to close that? I don't know why I'm getting that. So far, the successful compounds identified from the chemical screens were SR1 and UM171. And both of these compounds were known to expand stem cells significantly. And I will go briefly about these two compounds, and I will come back to them later. So both of these identifications were from a bigger compound screening platforms. They found aryl hydrocarbon receptor antagonist can promote expansion. The aryl hydrocarbon receptor is a cellular pathway for sensing some of this xenobiotics, and they found that inhibition of the pathway produces stem cell expansion. And then it comes another screening, they identified pyrimidindole derivatives. But if you see that the pyrimidindole derivatives expanded much better than the SR1 in terms of stem cell number. But the only caveat here was they don't know the molecular mechanism for that. So it becomes also a bit risky to use a compound without unknown molecular mechanism. But overall, these two compounds expand stem cells significantly over other discovered methods, and both of them are currently in clinical trials. Especially UM171 is in successfully finished phase 2 clinical trials, and it's showing a lot of promise. However, the molecular mechanism was not shown before, and a part of our work heavily involves UM171. So please keep an eye on this slide. Yeah. So my first part was hematopoietic stem cell identification. Although it was quite easy to say the stem cells sit in the apex of the hematopoietic hierarchy, identification of these cells are not so easy. using a set of cell surface markers, these hematopoietic stem cells are quite well defined in mouse models, but unfortunately, there is barely any overlap between the surface stem cell surface expression of surface markers between human and mouse. So, which makes it difficult to use the same surface markers for human cells. As a result, independent studies identified a set of stem cell markers for human hematopoietic stem cells. But even then, after using this 5 different markers, we can isolate like stem cells with a maximum 10% purity. So in reality, we don't have a set of surface marker to pinpoint and isolate the hematopoietic stem cell. And the issue even gets worse when we start expanding stem cells in culture. As I said, these 5 markers define stem cells, but during in vitro culture, 3 of these markers are also not reliable and they are not associated with stem cell activity anymore. So it makes it really difficult to identify a suitable stem cell marker in vitro culture. So the first part of my work was involved in identifying a novel stem cell marker to trace the stem cells in the in vitro culture. And the second part of my talk is going to be focused on a novel molecular target where we can expand stem cells for clinical use. So for the audience who are not from this background, I would also like to introduce a technical slide to show how we isolate hematopoietic stem cells, because which might be also a curious question for many. At first, we isolate the whole WBC from the cord blood or from the bone marrow. Then we use a cell surface marker called CD34, which is going to be used often after this slide, which says the stem and progenitor cells from the blood. Those are marked by CD34. So commonly we call the stem and progenitor cells as CD34 cells. And in this system, there is a nice technique to isolate these cells. We use an anti-CD34 antibody, which is coated with a magnetic bead. So once we add this antibody to the cell suspension, all the CD34 expressing cells are tagged with this antibody. Then we pass these cells through a column which is fixed on the magnet. As I said, these are some kind of iron beads. They are getting trapped in the column, whereas the unmarked cells, they will pass through the column. So after washing the column for a couple of times, once we take the column out of the magnet, and if we elute the column, what we get is the CD34 positive cells, which is the hematopoietic stem and progenitor cells. Normally, this population consists like 1% of the WBC cells, which is so rare, and using this method, we can enrich them to more than 90%. And then I also told about a set of other stem cell markers. If we are interested in using these markers, we can actually stain the cells with antibodies against the surface markers. Of course, these antibodies will be coated with fluorochromes with different colors. Then we can pass the labeled cells through flow cytometer, and in this flow cytometer setup, we can separate the populations based on the surface marker expression, and we could eventually sort out the desired subpopulation or subpopulations. It's quite easy. It's quite convenient. So these 2 techniques are indispensable for hematopoietic stem cell research and are used in day-to-day applications. So when I started this project, I was analyzing the gene expression data from the stem cells and the progenitors, especially stem cells from umbilical cord blood and fetal liver. And what I found was there is one gene called ProCR, which was significantly expressed at higher levels in stem cell populations compared to the progenitor populations. And ProCR encodes for a protein called endothelial protein C receptor, which is also called EPCR. So then we thought maybe EPCR is a reliable cell surface marker for human hematopoietic stem cells. So later we identified that hematopoietic stem cells from human fetal liver, they are exclusively marked by EPCR expression, as we can see that the mouse transplanted with fetal liver EPCR positive cells engrafted and produced blood cells, whereas the EPCR negative cells didn't produce any blood in mouse, which means all the stem cells are EPCR positive from the fetal liver. But in umbilical cord blood, what we found was some of the stem cells were expressing EPCR and some of the cells were not expressing EPCR. So maybe there was a partial loss of EPCR during this stage of human development. But surprisingly, what we found, which is quite relevant for this presentation or for our study, when we put stem cells from either fetal liver, bone marrow, or from cord blood, we always found the hematopoietic stem cells gaining EPCR expression, and EPCR was a really reliable stem cell surface marker for isolating or identifying stem cells in the in vitro cultures. So this made our next research possible. And this study was published in 2019 in Hematologica. Then my second part of the talk is going to be focused on a gene called lysine-specific demethylase, also called LSD1. So what is this lysine-specific demethylase? As you all know that the DNA is regulated epigenetically at multiple levels. Histone regulation is one of the major epigenetic regulation of gene expression, and histones usually have a tail which has a lot of lysine residues. They can be either methylated or acetylated, and which could produce either open the chromatin or resulting in closing the chromatin. As a result, we get gene expression on or off. So what does LSD1 does? LSD1 actually removes mono and dimethyl marks from the histone 3, K4 and K9 residues. So it's been previously reported that when LSD1 removes mono and dimethyl groups from the histone tail, as a result, the corresponding genes are silenced and they cannot express anymore. And it's also happened to be that most of these genes are hematopoietic stem cell identity associated genes. As a result, the expression of LSD1 always suppresses expansion of stem cells, and it promotes differentiation. So our question was, how can we block the differentiation for a few days? As a result, we can expand the stem cells in vitro. So to achieve that, our idea was to block the LSD1 with small molecule inhibitors, which is a transient inhibition that lasts for a few days, and cells will regain the LSD1 activity afterwards. But this few days of LSD1 inhibition could promote the expansion of stem cells. That was our hypothesis in this project. So to do that, we took human hematopoietic stem and progenitor cells and treated them with 3 different pharmacological inhibitors of LSD1, 2-PCPA, GSK-LSD1, and RN1. And we treated the cultures for 6 days, and then we did FACS analysis for EPCR and CD34. In here, basically the cell population which is positive for CD34 and EPCR is called hematopoietic stem cells, which is what we identified from our previous study. And of course, the population which is negative to EPCR, positive to CD34 is progenitor population, and the CD34 negative is more differentiated cells. So what did we observe after 6 days? We observed like a remarkable expansion of stem cells with the treatment of LSD1 inhibitors. As you can see, it's only 2% of the cells were retaining EPCR expression, whereas more than 25% in couple of LSD1 inhibitors, and with RN1 still we observed like a 5-fold expansion. So in conclusion, we observed that treating the stem cell cultures with LSD1 inhibitors promotes a rapid expansion of phenotypic stem cells. Then later we did a dose titration experiment to identify the ideal dose and ideal compound for the further studies. So we used 2 different readouts here. As I said, it's the stem and progenitor cells here, and it's mostly the stem cell population here. What we found was when we used 32 times less than IC50 concentration, the LSD1 inhibitors were producing predominant expansion of CD34 positive cells, and especially with 2-PCPA. So since this dose was expanding stem and progenitor cells ideally, and 2-PCPA was the best among the 3 inhibitors, we did all the subsequent studies with 2-PCPA. Then, once we successfully expanded the phenotypic stem cells, but we have to evaluate the functionality of the stem cells, because sometimes the surface marker expression could be nothing unless the stem cell is performing. So to achieve that, we did a quite laborious work, I would say, because this assay is called a limit dilution experiment for those who doesn't use this assay. So in this assay, we typically take 4 different cell doses, 5,000, 1,300, and 100, and each of this receives 10 mice, and of course this is for one condition DMSO. Then for the 2-PCPA also we did the same experiment, and of course this experiment was repeated as well. So altogether we have to use more than 200 mice, and the mice model we use for such studies are called NSG mice, because these normal mice will reject human cells due to the residual immune cells in the mouse. As a result, we have to use a special mouse model which is non-obese diabetic but also lacking IL-2 receptor expression, which means the mice lacks any adaptive immune cells, and this also has further mutations. As a result, it lacks complete development of immune cells. As a result, we can transplant human stem cells, and the mice can successfully repopulate human hematopoietic stem cells. And we usually leave the mice for 18 weeks because usually the transient cells we transplant, they disappear by 8 weeks, but the real stem cells can persist for 18 weeks and still be able to produce new cells. So the cells which are persistent 18 weeks post-transplantation means those are coming from the stem cell activity. By doing this Poisson calculation, we identified that indeed the number of stem cells are significantly, functional stem cells are significantly higher with LSD. Hello? I think I lost the network. I'm joining again. Yeah. But I need, yeah, I got it. Perfect. Yeah. So next, we mapped the early transcriptional changes induced by LSD1 inhibition. To do that, we sorted the pure HSC population and we treated them with either control, PMSO control, or 2-PCPA for 24 hours. Then we did the global gene expression profiling, and later to know that what kind of genes are induced by the LSD1 inhibition, we did GSE analysis. In this analysis, we have a gene set database which are having gene sets associated with different biological functions. So we used more than 10,000 different gene sets, and we run GSE analysis. And I will show two of the significant results here. A first result, what we found was 2-PCPA inhibition induces a perfect hematopoietic stem cell gene signature, which means it not only produces a phenotypic or functional effect. The genes induced by 2-PCPA are stem cell associated genes. Then, surprisingly, we also found the gene signature induced by 2-PCPA was quite similar to UM171. As I said before, UM171 is also another small molecule which is in clinical trials for stem cell expansion, but the molecular mechanism of this compound was not known. So we were quite excited because we might be closing to identify the molecular mechanism for a drug which is in clinical trials. So to summarize the data so far here, what we found, inhibition of LSD1 promotes the expansion of phenotypic stem cells. And of course, the LSD1 expanded stem cells were functionally superior, and the stem cell numbers were quite higher compared to the DMSO control. And later we also found LSD1 inhibition was inducing a gene signature quite similar to UM171. As I said, UM171 is a molecule in clinical trials without a known molecular mechanism. So later we thought maybe UM171 is also one of the LSD1 inhibitors, just inhibiting LSD1. But to test that hypothesis, what we did, we chose an in vitro assay. In this in vitro assay, we took a small peptide which is carrying dimethyl groups. So typically, when we use a purified LSD1 enzyme, it removes one of the methyl group from the dimethyl peptide. As a result, it releases hydrogen peroxide. The hydrogen peroxide can be used in combination with other compounds. As a result, we can read out the LSD1 activity as a fluorescence. So this kit was designed, and we used 2-PCPA, a known inhibitor of LSD1. And as you can see, a dose-dependent increase of 2-PCPA significantly inhibits the LSD1 activity, and no inhibitor control is here, and this is the background. So 2-PCPA inhibits the LSD1 as expected. However, though UM171 is producing a similar gene signature as LSD1 inhibition, but UM171 compound could not inhibit LSD1 in vitro. Then we thought the mechanism is not as simple as we thought. It might be quite complicated. Then we did further assays to validate that. As I said, LSD1 can be part of many different chromatin remodeling complexes like CoREST complex, NERD complex, ER alpha and androgen receptor, and it can also modulate two different activities. Then we thought maybe UM171 is targeting one of these components. As a result, it's expanding stem cells and providing a similar molecular signature like LSD1 inhibitors. To do that, we did a complex mass spectrometry assay. In this assay, the whole cellular proteome is treated with the compound, and the proteins in which the compound binds can have an altered melting temperature. How is to read out that altered melting temperature? In this assay, we treat quite a lot of cells and we aliquot them into multiple vials, either control and are treated. Then we heat this cellular lysate to different degrees in a gradient from 37 to 67. So usually most of the cellular proteins tend to melt in between this temperature. But if the drug is binding to a particular target protein, only that target protein will melt at a different time point. So usually the denatured proteins will be pelleted and the remaining proteins will be in the supernatant, and those supernatant will be collected and analyzed by mass spec. With this assay, we can identify the targets of small molecules which are not known. With this assay, we actually choose two different cell lines to perform this assay, because this cell line this assay demands 100 million cells, which is quite difficult to isolate from cord blood. We have to isolate for maybe 6 months, which is not worth to invest that many cells here. So we used two different AML cell lines, which is also derived from blood cancer. So we thought it's quite convenient to use the cell lines. And we filtered out the candidates identified from the mass spectrometry. We got nearly 130 candidates from HL60, and we got 44 candidates from Cassimi-1 after applying our filter, where the protein should show more than 1 degree melting difference change, and it has to perform consistently between the replicates. As a result, we identified 5 different candidates, and one of them was RCOR1, which is a core member of the CoREST complex, which is also binding with LSD1. Then boom, we got the target, which is the RCOR1. Maybe that's where UM171 binds and destroys it, and as a result, it modulates LSD1 activity. However, as I said, none of these genes are altered when we treat UM171 at the transcript level. So mostly the destabilization happens at the protein level. So to test that, we treated human hematopoietic stem and progenitor cells with 2-PCPA and UM171, and then we probed for all the members of this CoREST complex. It was so easy. Then we found, as expected, treating with UM171 completely destroyed the CoREST complex, mainly RCOR1 and LSD1, and to some extent HDAC2. So this study confirms that the small molecule UM171 also targets targets LSD1 and RCOR1 and destroys it for degradation. Then, as I said before, LSD1 can modulate either K9 methylation or K4 methylation. So we were interested to demonstrate what kind of methylation is affected here. To do that, we treated the cells with LSD1 inhibitor and UM171 for 24 hours. Then we did a Western blotting for global K4 dimethyl marks and global K9 dimethyl marks. And what we found was we found significant increase in global K4 dimethyl marks with 2-PCPA as well as UM171 treatment. And there was no involvement of K9 dimethyl marks. So which again confirms that the degradation of CoREST results in the accumulation of dimethyl marks, and that's how it drives the hematopoietic stem cell genes. Then we want to confirm again by inhibiting RCOR1 function, but unlike LSD1, which has a pharmacological inhibitors, which has also an enzymatic activity, RCOR1 is a scaffold protein. It doesn't have any active sites where we can inhibit with small molecules. But then we have CRISPR. So for those who doesn't use CRISPR, I would say it's a guide-directed DNA endonuclease. It cleaves the DNA. As a result, the cleaved DNA can be repaired, but the repair process is not as efficient. As a result, it introduces point mutations or frame shift mutations, which destroys the gene function. So we used this approach to target ARCOR1. So how did we achieve that? So we transduced human hematopoietic stem and progenitor cells with lentivirus expressing ARCOR1 guides. This is the guide RNA. But then we introduced the Cas9 protein as an mRNA through electroporation. As a result, the delivered mRNA receives become Cas9 protein, and it receives the guide which is expressed inside the CD34 cells, and it can cleave the target gene. And we did Western blotting. As you can see, with 3 different guides, we successfully eliminated ARCOR1 protein expression. And as expected, the loss of ARCOR1 promoted a sharp expansion of hematopoietic stem cells, so which again proves the fact targeting either LSD1 or the associated ARCOR1 protein promotes stem cell expansion for functional stem cells. Then we also quantified with the replicates and with the repeats. We find like these guides can promote like 3 to 8-fold expansion of stem and progenitor cells, and it also expands nearly 4 to 8-fold expansion of phenotypic stem cells. So altogether, so far we proved that targeting COREST complex expands functional hematopoietic stem cells, either by inhibiting LSD1 or by destroying ARCOR1. So this is what we have found so far with small molecule UM171, and we also wanted to see, since the previous studies were done 24 hours post-treatment, we wanted to see even if the drug can degrade protein like earlier than 24 hours. So we did a time kinetics experiment. We chose 1 hour, 3 hours, 6 hours, and 24 hours. And we did Western for ARCOR1 and LSD1. And we chose cell lines for these studies. We chose 3 different cell lines. As you can see, the panel represented here is for 1 hour, 3 hours, 6 hours, and 24 hours, either with or without drug. As you can see, at 1 hour, there is no change in the ARCOR1 and LSD1 protein levels. But at 3 hours, we started seeing the reduction of ARCOR1 and LSD1, and the degradation was predominant by 6 hours. And we could observe similar trend with other 2 cell lines, HL60 and U937 as well. So this clearly establishes that the small molecule treatment somehow induces the protein degradation as early as 3 hours, which is quite interesting to investigate further. Then I would also like to put a smaller pathway here, how the endogenous proteins are getting degraded. Since we want to understand this mechanism more, there are 2 major pathways of cellular protein degradation. The first one is called a ubiquitin proteasomal system. In this system uses a protein called ubiquitin, which is a quite small protein like 8 kD. And there is a set of ligases which can add ubiquitin to the target protein. Typically, the target protein receives multiple repeats of ubiquitin, sometimes even branched chains of ubiquitin. As a result, the polyubiquitin chains are recognized by proteasome, which is the black hole inside the cell. It just swallows anything polyubiquitinated, and it destroys the cellular protein and recycles the amino acids. It's a simple pathway compared to autophagy. Autophagy, in other way, it is it has 3 sub pathways, but altogether the concept here is the protein is targeted towards lysosome for degradation. So the enzymes in the lysosome degrades the target protein and recycles it. So we wanted to know the degradation of ARCOR1 and LSD1. Does it happen through ubiquitin proteasomal system or autophagy? At first, we wanted to test the ubiquitin proteasomal system. For that, we treated the cells either with UM171 or UM171 along with bortezomib. It's a blocker of proteasomal activity. So the idea here is if we block proteasome, we would prevent the degradation if the degradation is happening through the ubiquitin proteasomal system. So as shown in this figure clearly, the increased doses of bortezomib prevented the degradation of ARCOR1 and LSD1, which clearly states that the degradation induced by UM171 predominantly happens through ubiquitin proteasomal system. So, as I said before, I would just extend a bit about the ubiquitin proteasomal system. As you can see here, it involves three major enzymes. The first enzyme captures free ubiquitin, activates it, and the enzyme 2 receives the ubiquitin from enzyme 1 and it transfers to the target with the help of E3 ligase. And this process is repeated over and over. As a result, the target substrate receives polyubiquitination and it is received by the proteasome, and eventually they are degraded. So then to show it convincingly, we wanted to show this the polyubiquitination happens to our target proteins ARCOR1 and LSD1. To do that, we made a setup here because there was not a good antibody to pull down ARCOR1. So what we did, we expressed ARCOR1 in a lentivirus with a 3x flag tag, and we transduced these cells to the cell lines. Of course, these were designed to express GFP, so we can sort out the positive cells and enrich the cells. Then we also tested if the overexpressing protein is also responding to the drug. So, of course, at first the constructor was expressing. As we can see, when probing with the flag, we observed the expression of the flag. Then when we treated UM171, we observed the degradation of the exogenous construct we expressed. So which means the construct is responding and it can be used for polyubiquitination analysis. And we treated these cells 2 hours and we did flag immunoprecipitation, and we also did LSD1 immunoprecipitation. We enriched these proteins by immunoprecipitation, and we probed with antibody targeting polyubiquitin chains. As you can see in the control and DMSO, there is no background signal, but when we pull down the flag with UM171 treatment, we observed a streak of polyubiquitin chains on the ARCOR1 protein, which says that of course the ARCOR1 protein is polyubiquitinated. Then we went on to demonstrate the similar polyubiquitination mechanism with LSD1 protein as well. As you can see, lower panel shows LSD1 is enriched and the upper panel shows polyubiquitination with UM171 treatment. And the bands are not so sharp here because the length of polyubiquitin chains can vary between different molecules of protein. So as a result, we always will see a streak. So I'm coming to the end of the talk here, and to put our overall study so far here, in the control conditions in the in vitro culture, the stem cells lose their methyl marks in the stem cell specific genes due to the chorus complex. It's mainly due to LSD1. As a result, the stem cell genes are silenced and they differentiate. But when we block the activity of this component by inhibiting LSD1, we observe like persistent methyl marks in these genes, and it also promotes stem cell expansion. And while we also used this small molecule UM171, which is in clinical trials without a known molecular mechanism, and we found that it also induces the degradation of the chorus complex, which also results in the similar outcome as LSD1 inhibition, and it promotes expansion. But what was this study? So what was the significance of this study? In here, we saw that blocking an epigenetic regulator, we can expand the functional stem cells. But more importantly, we are showing the molecular mechanism for a small molecule drug, which is in phase 2 clinical trials. And the details of this study are presented in this publication in the Blood Journal, which came quite recently, like a month back exactly. So you can you're free to visit this page and you can get more details. It's an open article, so anyone can download and read the whole story there. Whatever I got presented included maybe 20 to 25% of the data. And in summary, we showed EPCR as a reliable cell surface marker for hematopoietic stem cells, and we also going on to show that LSD1 inhibition expands hematopoietic stem cells. And of course, we also went on to demonstrate that LSD1 containing core-rest complex is the main target of stem cell expanding clinical molecule UM171. With this, I would like to wind up my talk, and I would like to acknowledge the people involved here. I would like to acknowledge my PI, Jonas Larsson, for his critical suggestions and encouraging me to pursue with this project, and also the other lab members involved in this project, and also the other members from the department and other members, especially Roman Zubarev from the Karolinska Institute and his group, and also Matthias and Kesian for helping with some of the samples here. And I would also like to thank FACSCore and the Animal Core facilities. And last, I would also like to acknowledge my funding sources, Swedish Cancer Society, for supporting me to pursue my postdoctoral research, and also the Leukemia Research Award from Lady Data Memorial Trust, and the grants from Physiographen. And also I would like to acknowledge Lund Stem Cell Center for all the support to pursue the study. That's all. Thanks everyone for joining. And yeah, I would like to take any questions. If some of you have some questions, please let me know.
So thank you, Dr. Agadeshwaran. Our participants has raised many questions.
Yeah.
So out of which a few questions were selected by us, and we would like to tell you the questions and give your good answers for them. So the first question is from a participant called Satya Vradu. So his question is, what are the precautions that we have to follow to avoid leukemia? So this is the first question raised by Satya.
So that's a $1 million question, because if everyone knows that, probably nobody will be getting leukemia. So honestly, like there are certain things one can follow to escape from the leukemia. Like you can avoid yourself exposing to any harmful radiation, and you can probably avoid yourself to xenobiotics, which affects DNA replication mostly, because there are a lot of xenobiotics which can affect the DNA replication and introduce mutations. So if you're cautious about it, you can avoid processing food to avoid this extra chemicals, and yeah, you can avoid all all these xenobiotics as much as you can.
Okay, thank you. The second question is from Krishna G. Nair. He asked, how does a same stem cell differentiate into different type of cells like blood cells and liver cells, etc.?
Usually that happens in the earlier stage of development. Those are called pluripotent stem cells. But the stem cells I was presenting here are called hematopoietic stem cells. These are called oligopotent stem cells. These pluripotent stem cells, they can produce any lineage in the body. These are typically present in the blastocyst stage. But once the baby develops, then after delivery, mostly what we find is oligopotent stem cells. They stay in the tissues and replenish the tissues for lifelong. So blood stem cells, they can only produce blood lineages as far as known. As far as I know, I don't think they can produce different other lineages unless they are reprogrammed to do so.
Okay, thank you. The third question is, how does the antibody interacts with magnet?
The antibody doesn't interact with the magnet. The antibody only interacts with the cell surface marker. But the other end of the antibody is connected with an iron particle, iron beads. So they iron nanoparticles to be precise. These nanoparticles can respond to the magnetic field. So as a result, they are getting trapped in the column.
Okay. So the next question is, this question is from R. Kavita. Her question is, can we use umbilical cord stem cells taken from the cord blood of one mother to treat many patients? Is there any specificity?
To date, I think with the HLA matching, it's possible to use from any donor to quite a few number of people. But what is lacking is the enough cell dose. So basically, if you preserve the umbilical cord blood stem cells, it can be used only to treat another kids, like maybe 4 to 5 kilograms of body weight. But if you want to use it for an adult patient or an adolescent, you have to expand them to a number which can be used. Because if you ask clinicians, they could give you the exact cell dose numbers. I'm still a researcher, but I can still say that you need to expand them quite a bit. And there is no way to maintain. Yeah, there is no way to maintain these cells forever in the culture. They will lose functionality. So we can only expand them for a limited period of time.
Okay, thank you, sir. The final question.
Yeah.
It's a hypothetical question. It is raised by a participant named Christina Sharon. Her question is people traveling in closed window cars for longer hours, are they prone to cancer?
I don't know. To answer that question, one has to do a research. Probably you have to recruit a group of people traveling in the car, a group of people not traveling, then you can compare the leukemia incidence. That's probably where she can get the answer. I cannot give hypothetical answers.
Thank you. So the question session has ended. Now over to Dr. Francis area. Thank you, Dr. Agdeshwaran. Thank you.
Thank you.
Thank you very much, Dr. Agdeshwaran, for your excellent presentation on your given topic. Your research work on the stem cell is very really exemplary. Your talk was an eye-opener session to all the participants.
Francis, sir.
Hello, Edward, sir. Hello. Hello. Hello, Francis, sir. Hello, Francis, sir. Hello, Francis, sir. Edward, sir. Can you hear me?
Sir, yes, I can hear you. Sir, I have informed the Francis. Dr. Francis, I have informed them.
I think, yes, I have a problem. Otherwise, why don't you continue?
Sir, sorry, could you repeat it?
Edward, sir, if Francis, sir, is not available.
Sir, our father was the president, sir. Or the secretary was the president.
Oh,
I'll
okay.
We have a problem in fixing mobile. For time being, let me access through mobile only for that. They have a problem in fixing. I find it difficult in fixing. Zoom further. That is easy. It is eStocks. It is another platform. A completely different platform. But I don't know what is. இங்கே வச்சிருவோம் ஃபாதர். கண்டினியூஸா வரும் ஃபாதர்.
So on behalf of this 3 days international webinar series, I am Dr. A. Edward, one of the organizing secretary of this webinar series. I is requesting Reverend Father Secretary to give you a benediction. Over to Reverend Father Secretary. Thank you.
Good evening to all of you. Dear professors, dear organizers, and my dear students, and the participants of this webinar. Good evening to all of you.
Good evening, Father.
The PG and Research Department of Botany and Department of Biotechnology of St. Joseph's College jointly organized a 3-day international webinar series on the theme Unlocking Approaches in Biological Research. At the outset, I congratulate the Department of Botany and Biotechnology for organizing such a useful and relevant webinar, a very useful webinar, I should say. I thank the organizing secretaries, Dr. A. Edward and Dr. T. Francis Xavier, for organizing at this moment COVID-19 at an appropriate time on an appropriate topic. Rather than allowing their mind to rust, the entire team have taken pains to share their knowledge on the scientific research and endeavor, discovering the relevant principles and using those principles to manipulate. I don't know whether we can use this word manipulate at this juncture. Manipulate biological system to meet one needs is the goal and objective of the growing field of biological research. As I listen to the webinar talks, the strategic interest of biology-driven research is the priority for the webinar. The researchers and the organizing secretaries have taken a lot of...
Hello. Hello.
Sir.
Dr. Edward,
Yes,
are you hearing me?
yes, sir.
Yes, sir.
Yes. So due to the technical issue, Dr. Father Secretary has joined now. So from my link, that is why it is disconnected.
Okay.
Sorry. But he already there. Half of the way.
Yeah.
He's in half of the way. Yeah. Already started speaking.
I think
As I listen,
Yes.
as I listen to the webinar talks, the strategic interest of biology-driven research is the priority for the webinar. The researchers and the organizing secretaries have taken a lot of efforts to bring together the scientists and researchers from various disciplines and helped them to share their experience, expertise, knowledge, energy, and enthusiasm. The webinar series has provided an opportunity for the multidisciplinary networking and knowledge exchange. I thank the organizers on behalf
அப்படியா? இப்ப நான் உள்ள வர முடியாதோ?
அதாவது ஃப்ரெண்ட் சர்க்கிள் நீங்களே பேசுறாருன்னா ஃப்ரெண்ட்ஸை கொஞ்சம் வெயிட் பண்ணணும். sir. The father is speaking in the Francis are being.
Thank the organizers for their efforts and appreciate their human service to scientific researchers. I wish the Lord's Almighty's choicest blessings and wish that they undertake many more useful research-oriented webinars. All the best. God bless you all.
Thank you, Father.
Thank you.
Thank you, Father. Okay, now we have entered the vote of thanks, and I am thanking Reverend for the Secretary for his wonderful valediction for this international webinar series. Thank you, Father. Thank you very much. So now the vote of thanks. Gratitude. Gratitude is not only the greatest of virtues, but the parent of all the others. Showing gratitude is one of the simplest yet most powerful things humans can do for each other. Good evening to all. And Dr. A. Edward, Head Department of Biotechnology, and one of the organizing secretary of this 3 days international webinar series. It is a great privilege and a great joy for me to extend a vote of thanks to all those who have contributed in one way or other to make this webinar series a success. First of all, I would like to thank our Reverend Father Rector for his blessings to conduct this international webinar series. Thanks to you, Father. Next, I would like to thank Reverend Father Secretary, who is always instrumental in encouraging and supporting us to conduct this type of programs for the betterment of humankind. He readily accepted our request to give you a valedictory oration, even in his hectic schedule, to be with us today. Thank you, Reverend Father Secretary. And great thanks to Reverend Father Principal for giving us the permission to conduct this international webinar series with the continuous encouragement and infrastructure. When we requested him to conduct the international webinar series, our Reverend Father Principal has immediately accepted and request and showed the green signal, go ahead. So this is the positive energy which we all receive from him when we approach him. Thank you, Reverend Father Principal. I'm also very thankful to the Deputy Principal, Dr. Alex Ramani. He is always readily available personality and helpful in many occasions. I thank him for the felicitation that he gave us on the first day of program. Thank you, sir. The 3 resource persons have brought here with them and shared with us their knowledge and experiences. On behalf of the entire institute, I would like to thank the first speaker, Dr. Joseph Selvin, Professor and Head Department of Microbiology, Pondicherry University, for his wonderful talk on metagenomics. His presentation was wonderful and it is very easy to understand. Next, I would like to thank our alumnus, Dr. M. Padmanabhan Moganan, postdoctoral fellow, Kwanghi University, South Korea, and he also did his MSc Biotechnology in our college, has presented a wonderful topic on genes and research with a lot of interesting points. And thanks to our alumnus, Dr. Agadishwaran, who is today's speaker, and he's the associate researcher in the Lund University, Sweden, for his wonderful talk on blood forming cells, which are very impressive, informative, and productive. We understand that all of you have taken out of your very important schedules to contribute to this webinar series. Thanks to all of you for your invaluable contribution. Next, I would like to thank Dr. Sagai Satish, Dean School of Biological Sciences, and Dr. S.R. Sendil Kumar, HOD Botany Department, for always being supportive in our efforts. Great thanks to Dr. Kanike Raj for his support and timing help in organizing the webinar series through Easy Talks and YouTube video links. Whenever we contacted him for our webinar, with his pleasant smile, he arranged everything needed for the smooth conduct of this webinar series. Thanks to you, sir. And great thanks to Dr. D. Francis Xavier, one of the organizing secretary of this international webinar series, in meticulously planned and organized the webinar along with us. He is always friendly and interactive professor. Thanks to Dr. D. Francis Xavier. Also thanks to our biotechnology staff, and thanks to our sister department, biochemistry, for the continuous support and encouragement, especially Professor Anthony Divakar, head of the department. I also wish to thank the participants, especially the professors, researchers, research scholars, and students who has participated from different districts of Tamil Nadu, other states of India, and other countries. Even in their tight schedule, all of them has regularly attended this 3 days webinar series and provided a lot of suggestions for the better conduct of webinar in the near future. I am sure that the participants must have been benefited by attending this webinar, and a final thanks to the persons who have worked extremely hard both in the foreground and background in accomplishing this webinar series a grand success. Last but not least, we all express our sincere thanks to our Almighty God for blessed this webinar series. Thanking all of you. Thanks a lot.
Thank you very much, sir. Thank you.
Thank you. Francis, sir. Francis, sir. The program has ended. Thanks to all of you for your active participation in this 3 days international webinar series. Thanking all of you. Thanks a lot. எத்தனை தடவை